We all have DNA, a unique genetic code that forms the basis of life. Comprised of nucleotide sequences bound into a double helix, DNA provides the instructions for each cell in our body. But what happens when something goes wrong in our DNA, and how can we fix it? Today we're going to dive deep into nucleic acid-based therapies, specifically into the use of antisense oligonucleotides to reduce plasma triglyceride levels, and how this can be used to treat a rare genetic disorder. It's sure to be an exciting episode today. Welcome to the genetic basis of stuff and things. Elevated triglyceride levels are one of the most common health problems in modern society. Whilst they can be broken down and used as an energy source, significantly elevated triglycerides increases the risk of developing cardiovascular diseases. Along with cholesterol, triglycerides travel through the bloodstream bound by lipoproteins, such as very low density lipoprotein synthesized by the liver and chylomicronemia synthesized by the intestine. Typically, elevated triglyceride levels are treated by quitting smoking, eating a healthy diet, and engaging in physical activity. However, some individuals have a genetic mutation which cannot be treated using these methods. Let's unpack a novel treatment to help these patients. A common mechanism for treating genetic mutations involves gene replacement therapy, in which a viral vector is used to deliver a functional gene into a mutated cell to restore the genome. Recall that DNA is transcribed into messenger RNA, which is translated into amino acids to form proteins. Therefore, over the last few decades, research has shifted to focus on using nucleic acid-based therapies to target RNA in order to alter gene expression. This is achieved using antisense oligonucleotides. These are short synthetic polymers of monomeric nucleotides which bind to a complementary mRNA region, inhibiting translation of the sequence and protein synthesis. Antisense oligonucleotides can be classified into three different generations, with modifications to the phosphate backbone or ribose sugar, or the addition of unnatural bases. Unlike previous technologies which alter the DNA sequence, antisense oligonucleotides simply alter gene expression, conferring reversible and precise advantages. Now that we understand the mechanism of action, let's discuss a specific treatment. In 2013, Graham and colleagues synthesized an antisense oligonucleotide sequence known as ISIS-308-401, which aimed to reduce plasma triglyceride levels. As a complex polygenic trait, the genetic basis governing plasma triglyceride concentration involves a variety of interacting genes, one of which has been identified as coding for apolipoprotein C3, or apoC-III, which is embedded in the outer membrane of lipoproteins. apoC-III is a glycoprotein consisting of 79 amino acids, primarily synthesized by the liver and functions to increase triglyceride levels through two mechanisms. Firstly, by inhibiting the activity of lipoprotein lipase, which breaks down the lipoproteins. apoC-III also reduces the binding of lipoproteins to hepatic receptors, preventing their uptake and metabolism by hepatic lipase. To target the transcribed 500 base pair apoC-III gene located on chromosome 11q23, Graham and colleagues synthesized 350 different second-generation antisense oligonucleotides, roughly 20 nucleotides in length, to target 200 different sites. 26 were further screened in transgenic mice, with ISIS-308-401 providing the greatest tolerability and accuracy. Before this, they also tested the effects of apoC-III inhibition in rodents using a rodent-specific antisense oligonucleotide sequence to confirm this desired result before administering ISIS-308-401 into non-human primates with conserved homology. Reductions on apoC-III and triglyceride levels were observed, so Graham and colleagues moved to testing in humans. Using complementary base pairing, this sequence binds to bases 489 to 508 of the transcribed apoC-III mRNA. This forms a DNA-RNA heteroduplex that activates RNase H1, which acts like molecular scissors to degrade the apoC-III mRNA, inhibiting translation and the formation of the apoC-III protein. With a reduction of apoC-III, lipoproteins can be broken down and cleared, reducing plasma triglyceride concentration. In this Phase 1 clinical trial conducted by Graham and colleagues, subcutaneously administered ISIS-308-401 provided dose-dependent reductions in apoC-III and triglycerides in healthy participants, as well as increased the synthesis of good lipoproteins and enhanced triglyceride clearance following meals. Doses were given once a week for 22 days, with the results being maintained for roughly 4 weeks following the last dose. Following this success, in 2019, this antisense oligonucleotide sequence, medically known as Volanesorsen, became approved by the European Medicines Agency to treat individuals with familial chylomicronemia syndrome. FCS is a rare autosomal recessive disorder, often caused by a loss-of-function mutation on the gene encoding lipoprotein lipase, located on chromosome 8p22. In the absence of LPL, triglyceride-rich lipoproteins accumulate instead of being broken down, causing severe hypertriglyceridemia, chronic pancreatitis, and reduced life expectancy, with the only current treatment being abstaining from high-fat foods, Volanesorsen provides an alternate method for triglyceride metabolism. As patients lack lipoprotein lipase, Volanesorsen primarily acts through the LPL-independent pathway by promoting the degradation of apoC-III mRNA by RNase H1, allowing the uptake and metabolism of lipoprotein in the liver by hepatic lipase. However, 76% of patients develop thrombocytopenia, a condition characterized by extremely low platelets. This has limited Volanesorsen's FDA approval, and therefore, further modification is required to ensure safety as well as efficacy. Overall, this is just one example of using RNA-based therapy to treat a debilitating disorder. But what other possibilities are there? With the ability of nucleic acid therapies to alter gene expression in contrast to editing the genome, this provides a promising avenue for the treatment of a plethora of diseases in order to give patients a second chance at life.